It's a real pleasure to be here, and I'd like to thank the organizers for the invitation to visit Bergen. I have to say that my initial reaction was somewhat skeptical. DigitalLife did not mean anything to me concrete, and my response was not perhaps atypical. At dinner last night, I asked the people at the table what they thought of as DigitalLife, and perhaps the most interesting response was the digital pet craze that spread across the world 20 years ago, which is perhaps not so unsurprising because the slogan of this is Tamagotchi life. So my work is on protein design, on enzyme design, and Gunnar, who had sent the invitation, explained that there is a very strong component of synthetic biology here. And the kinds of molecules that we're trying to create could be useful in that context. So enzymes are really amazing catalysts. They make life possible. They're responsible for accelerating the thousands of transformations that occur all the time in every cell on the planet. They're also being used increasingly outside of their normal biological context, sometimes on very large scale in factories. But what do you do if nature has not invented a catalyst for the reaction that you would like to accelerate? What do you do? And there are a number of possible strategies to address this issue. And I think one of the most promising combines computation and evolution. Design is really hard of an enzyme because these are incredibly complicated molecules, thousands of interactions that are organized spatially and temporally that allow very efficient conversion of substrates into products. The computational approach is illustrated here on the first slide. You choose a reaction that you would like to catalyze, and then using your understanding of the physical chemical properties of that transformation, you identify the rate-determining step, which needs to be stabilized preferentially in order to see catalysis. And then using advanced quantum mechanical methods, you can calculate the structure of that species, which is shown here in the kidney bean. In the transition state, you have partial charges developing, partial bond lengths, and you need to stabilize these. And in silico, you can do that by providing complementary interactions. So for example, if in the transition state, you develop a partial positive charge, you can provide an acetate. Place it properly to get maximal electrostatic stabilization of that entity. If you have a partial negative charge, you can provide a cation, a butylamine, to mimic the side chain of a lysine, for example. You can have a hydrogen bonding interaction, etc. And this collection of transition state structure and catalytic residues represents a mechanistic hypothesis. This is how we think we'll be able to accelerate the reaction, and the quantum mechanics tells us by what order of magnitude that should be possible. This is what Ken Houck calls a thiazyme. This is an idealized active site, and because it's in silico, you can dock it into the structure of any structurally characterized protein in the PDB. There are tens of thousands of candidate molecules that can be used for this purpose. We've collaborated with several groups in the United States, David Baker most prominently at the University of Washington in Seattle, and also with Steve Mayo at Caltech. They've developed programs like Rosetta Match that facilitate the search for a sterically complementary home for the thiazyme. And what's important is that the thiazyme not just fit within a cavity in the pocket, but that you be able to attach the catalytic groups to the polypeptide backbone. So for example, if we're using an acetate to stabilize a cation, then C2 of that acetate molecule has to be attachable to one of the C-alpha carbons in the polypeptide chain. Now this is just an initial hit. Now we have to optimally pack the active site. We have to make stabilizing interactions with the transition state, and we also have to orient the catalytic residues. And this is done, for example, with another Baker program called Rosetta Design, which considers all 20 amino acids at one site against all 20 amino acids at another site, considering all of the rotatable bond possibilities. It chooses the right combination the one that gives the most favorable energies, and then moves iteratively through the entire binding pocket. Typically, you sample 10 to 12 different residues, and you can cover 10 to the 20th different possibilities of residue identity and conformational preference. This vastly exceeds anything that we can do experimentally in the laboratory. And that's the real advantage of computation, because it allows us to navigate regions of sequence space that would be otherwise inaccessible to experiment. In the end, the computational programs give you a ranked list of candidate designs, and you produce some of them and test whether or not they're active. And I just illustrate this with one example from our own lab. This is a project that was carried out together with Steve Mayo at Caltech. The idea was to create a catalyst that would be able to do a simple deprotonation reaction. And the transition state molecule is shown in green here. This is a benzysoxazole derivative undergoing a CH hydrogen abstraction reaction. And in the thiazyme, we have a carboxylate as a base. We have a group to protonate the developing negative charge, and then we have a stacking interaction. So 3 catalytic residues, as it were. These are then modeled into a variety of protein binding sites. In this particular example, a Tim barrel, which is very commonly observed in enzyme folds. The starting protein is then produced in the laboratory, tested for its activity. Turns out in this particular case, the activity was not very high, but then we can use laboratory evolution, iterative rounds of mutagenesis and screening to gradually but continually increase the activity. And in the end, we have a catalyst that we can characterize biochemically. In X-ray, we can get high-resolution X-ray structure. In this particular case, you can see the carboxylate back here interacting with an inhibitor. The molecule is actually bound in a different orientation than was designed, hinting at problems with the accuracy of the original predictions. The group that was intended to stabilize the developing negative charge is no longer playing that role in the optimized catalyst. Instead, a new catalytic residue emerged in the course of evolution, teaching us something about the preferences of this particular binding pocket for achieving efficient catalysis. In the end, though, this is the transformation that was catalyzed, we were able to improve its activity, the starting computational design, by 1,000-fold, generating a catalyst that could do 700 turnovers of substrate per second per active site. That's an incredibly high efficiency. It corresponds to a rate acceleration nearly 1 billion-fold over the spontaneous transformation. Very similar to the kinds of activities we associate with natural enzymes. And this technology has been applied to many other transformations, abiological Diels-Alder reactions, hydrolysis transformations, and a multi-step reversible aldol condensation reaction. In general, the starting activities straight out of the computer are modest, but through this iterative process of laboratory evolution, it's possible to improve their properties significantly. As I told you, here 1,000-fold, here 100-fold, a little bit less, and almost 10 to the 5th fold. So in the best cases, we really can make enzymes that exhibit the reaction rates and the selectivities we associate with true natural enzymes straight out of metabolism. As we look forward, though, we need to be able to move beyond relatively simple chemical transformations like the ones you see illustrated on this slide. And to do so, one will need to exploit other chemical features, particularly cofactors, to access regions of sequence of reaction space that you can't just access with simple amino acid side chains. In this context, metal-ligands are going to be particularly important. It's estimated that 40 to 50% of all natural enzymes utilize metal-ligands. And these metal-ligands are responsible for some of the most amazing chemistry that nature accelerates. And this extends from ripping hydrogens out of unactivated CH bonds, polymerization of nucleic acids, the reversible oxidation of molecular hydrogen, nitrogen reduction, and a host of other metabolic transformations. If you'd like to design an enzyme, what is a metalloenzyme? What is really essential? Well, we need the metal-ligand, We have to choose a proper scaffold to house it, and we have to figure out some way of anchoring the metal in the binding pocket. And then lastly, we need to figure out how to bring a substrate close to the metal ion using non-covalent interactions to force it in proximity to the metal so that it can convert the substrate into a product. Now, there are many strategies for designing metalloproteins, and some of the actors in the world who have contributed to this area are listed here at the bottom of the slide. Excuse me. In nature, metalloenzymes are believed to have arisen from much simpler peptidic precursors. So you can imagine that you have that in nature, you generate a small peptide, and then by a process of dimerization, you can generate a much larger structure. And that dimerization can, in principle, be facilitated by a metal cofactor. So it can coordinate groups and bring these peptides together. And then at the genetic level, the peptides could be linked through a flexible segment, and then that peptide folded to give you an active catalyst. So we can migrate from a prosthetic group, a structural prosthesis, to a catalytic feature. And we wondered whether we could duplicate this process in the laboratory. In fact, the first steps had been undertaken by Brian Kuhlmann, with whom we've collaborated some years ago. He took a 46 amino acid helix-turn-helix motif, which is shown here, and then using the Baker program, Rosetta Match, redesigned the upper surface of that molecule so that it could be self-complementary. And in that process, he introduced 4 histidines, 2 for each of the helical chains, so that in the presence of a zinc ion, you would get dimerization. And he was able to, and this protein he called MID1, Metal Interface Design Number 1. And so you see the design here. He actually produced the protein, which turns out to be homodynamic, very, very stable. And the crystal structure shows that the design algorithm gave him a molecule that's actually quite reasonable in terms of its structure. So the overall crossover angle of the 2 helix-turn-helix motifs are quite accurate. There is, however, one major deviation from the prediction. The idea originally was to coordinate each zinc with 4 histidines, 2 from each chain. And you can see in the crystal structure that the zincs are only held in place by 3 of the histidines, and 1 of them is turned away. In fact, the 2 that are turned away make a hydrophobic contact in the middle of the protein. That's interesting. It suggests that the computational predictions are not fully accurate, but it's also potentially useful because it means that you have an open coordination site for ligands. And in the crystallization buffer, there was a tartrate molecule that occupies this pocket. And so you can imagine replacing that tartrate molecule with a potential substrate. So for example, you could put in an activated ester. There are many zinc hydrolyses that have this kind of geometry. And Brian was able to show that he could hydrolyze activated paranitrophenyl esters by a zinc-bound hydroxide. The rate accelerations were not very high, but they're sufficient for evolution. And so we wondered whether we would be able to take this starting homodimeric metallopeptide and turn it into a globular metalloenzyme. And the specific substrate that we chose was this coumarin ester. We chose it for 2 reasons. First, it's relatively large, so it has a very nice recognition surface. And secondly, it has a chiral center. So we could also look at the ability of the protein to recognize 2-handed substrates. The spontaneous reaction is relatively slow, 10 to the minus 5th per second. So to make the globular protein, we took the original MID1 design, and then we connected the 2 chains with a short polypeptide linker with a flexible glycine serine-rich sequence. And then, in order to be able to focus on a single zinc site, we removed this one computationally, replacing the 3 histidines with two hydrophobic residues and a lysine that could form a very tight interaction network. And you can see that as you do this, you don't lose very much in catalytic efficiency. So this is our globular starting point now for investigating activity. This protein does accelerate the target ester hydrolysis reaction. Here is a typical Michaelis-Menten plot that you're familiar with from your undergraduate studies. We're looking at initial rate versus substrate concentration. From this analysis, you can derive kinetic parameters, kcat over km. You can see it's a catalyst, but it's not a very good catalyst. Natural enzymes are many, many orders of magnitude faster than kcat over km of 14 per molar per second. But we can optimize this using the standard laboratory evolution tools. And this just shows you the standard work flow in our laboratory. We can take the gene for our starting construct, our globular metalloprotein. We can introduce mutations randomly, and there are many ways to do this. You can buy kits that allow you to focus on a single site, perhaps close to the metal binding pocket. You can sprinkle mutations throughout the entire length of the gene using error-prone methods. You can combine favorable mutations using shuffling methods. In the end, you create a library of gene candidates, and you use those to transform E. coli cells. Each cell gets 1 plasmid and can produce 1 protein. And if we grow these cells in arrayed fashion in microtiter plates, we don't use very high throughput. We have 96 well plates. We can separate the genotype and phenotype characteristics of these molecules. And we bust open the cells, add our fluorogenic substrate, and just look for signal. So we can do in 2 weeks about 1,000 variants. Sometimes we look at 100, sometimes we look at 1,000. It depends really the specific questions that we're asking. But again, this is not incredibly high throughput, but it's sufficient in order to optimize the properties of these molecules in a continuous fashion. So we took the metalloprotein starting point and subjected it to 9 rounds of mutagenesis and screening. And in that process, we introduced 18 mutations. And you can see that these are distributed throughout the entire molecule. Roughly half of them are in the top helix, turn helix motif. Roughly half are in the bottom. These are shown by the magenta balls. Because we've mutated almost 20% of this protein is only 94 amino acids in length. We also began to interrogate the metal coordination site. Has something changed? And so we did cassette mutagenesis, alanine scanning of the potential coordinating groups for the zinc ion. And what we found is, in fact, this histidine is not important. So we can mutate that to an alanine, without any change in activity, suggesting that there's another coordinating group. And in fact, the histidine that had switched away turns out to be the most important residue in the entire protein for activity, suggesting that over evolution, many different things have changed. However, cumulatively, these have improved the activity very, very dramatically. So by 70,000-fold. Our new kcat is 2 per second, so we're converting 2 substrate molecules per active site per second. Kcat over km is now almost 1 million per molar per second. So this is 2 orders of magnitude away from the diffusion limit, which is a very respectable number for an artificial enzyme designed in the laboratory and evolved over this relatively simple procedure. So we can put this into perspective using the same kind of mountain fitness landscape that Janine presented yesterday. So our starting metallopeptide is somewhere in the foothills. It's not very active. It accelerates the reaction by 3 orders of magnitude, but it's not really so special. Here we're looking, by the way, at rate acceleration versus apparent transition state affinity. So the activity is actually not so dissimilar from that of carbonic anhydrase, a natural zinc protein that is important for the hydration of CO2. It also has promiscuous esterase activity, which is what I'm showing here, and it's a little bit better than our starting design. However, we introduce mutations. In the first round, we open up a pocket to bind this larger ester substrate. We increase the activity a bit, After 4 additional rounds, we climb the mountain until our best variant is, again, 100 per molar, 1 million per molar per second in terms of catalytic efficiency. This can be compared with the computationally designed enzyme that relied on a cysteine histidine dyad to do chemistry. And this is after evolution to optimize the rate of acylation of the protein. This is a very inefficient turnover catalyst because deacylation turns out to be very, very slow. And you can see that using the metal ion allows us to overcome this intrinsic problem. So the metal ion's intrinsic reactivity provides us avenues to much more efficient chemistry than we could using the original computational design. The evolved catalyst is also highly enantioselective. It preferentially cleaves the S-configured Ester substrate. And the preference for S over R is about 1,000-fold. So it's a very, very high degree of molecular recognition. We have a crystal structure now of the catalyst bound to a phosphonate transition state analog, and you can see that structure here. And gray is the protein. It looks very much like a typical globular protein. That has a small cavity, which serves as the active site. Here you can see the zinc poking into the binding pocket, interacting with the phosphonate group of the inhibitor. So if we cut away everything, you can also see why this is such an enantioselective catalyst. You have an extremely snug fit to the phenylpropionate moiety, which is the most deeply buried part of this molecule. Here's the methyl group sitting up in the back. There's no room for a methyl group pointing down. So this tells you why this enzyme is able to distinguish between the left and right-handed forms of our substrate. We can also look at the interactions within the binding pocket. So here you can actually see the zinc coordinated with the 3 histidines. Note that one of the histidines has acquired an additional hydrogen bonding interaction with a residue over the course of evolution. This is very typical for natural zinc proteins, where peripheral interactions are used to tune the reactivity of the metal center. The transition state analog is bound via one of its oxygens. We have a tetrahedral anion to mimic the transition state of the reaction. The other oxygen is stabilized by this arginine via a bidentite interaction. This is again an interaction that appeared spontaneously in the course of optimization. Note too that the arginine makes a cation pi interaction with the coumarin leaving group and may explain why this is able to depart so efficiently from the active site. I told you that there were very dramatic changes in the nature of the structure, and you can see that in this morph movie. So this is the original mid-1 design. This is what happened in the course of evolution. We have this very major change in conformation in the crossover angle of the helices. The new helix is also bent because there's a proline that's been inserted, and that bend would be very difficult to predict using computational methods. But it's absolutely essential for the success of the design because it enabled creation of the large binding pocket that houses the substrate. So this gives you an indication of the magnitude of the difference. We have a 36-degree switch in conformation, highlighting the malleability of these helical bundle scaffolds. So this story shows you that it is possible to go in the laboratory in a practical, convenient, relatively rapid fashion from a small peptide to a large globular metalloprotein that's able to catalyze its target reaction with high rates and high selectivities. In principle, this should also have promiscuous activity. Janine mentioned yesterday the importance of exploiting promiscuous activities in natural systems. Well, artificial enzymes are a very good source of promiscuous activity. They have no evolutionary bias. So here we have a zinc site with an open coordination. Perhaps we can bind completely different molecules like this azochalcone and carry out reactions with it. For example, we could add a vinyl indole and promote an abiological Diels-Alder reaction, a 4+2 cycloaddition, to generate a dihydropyran. This is a very common motif in interesting natural products, and being able to synthesize these molecules enzymatically would be quite useful. For those of you who are not familiar, who are not chemists, Diels-Alder reactions are a great way of simultaneously making two bonds and setting multiple stereocenters in a single step. And that's a process that's not used very often by natural proteins. There are a couple of examples of enzymes that accelerate unimolecular versions of 4+2 cycloaddition, but there are no examples of proteins that are able to catalyze a bimolecular reaction like this. And certainly none of these enzymes use metal ions. So can we catalyze this kind of abiological transformation? The starting protein does not. And so we turn to our friends Ken Hauck and his postdoc, Gonzalo Jimenez, who used the Rosetta model to predict changes in the active site that would perhaps delimit the binding pocket a little bit better. And using Rosetta, they suggested introducing this tryptophan and this leucine on both sides of the pocket. And you can see that that would provide a space that would allow the substrates to bind in a productive orientation for effective reaction. In the event, putting those two changes into our scaffold gives us a catalyst. It's not a very good catalyst. So in yellow and orange here, you see the rate acceleration over the spontaneous transformation with just zinc in the buffer. But it's enough, again, to do evolution. And so here we carried out 7 rounds of mutagenesis and screening, exactly the same kind of procedure that I showed you before, and introduced 10 mutations into the scaffold. Those 10 mutations increased the activity of this catalyst by 590,000-fold. So 5 orders of magnitude. So a huge increase in activity for this unnatural transformation. The kinetic parameters are shown here in this Michaelis-Menten plot. So now we have an enzyme that will do this reaction with 11 turnovers per second. The Michaelis constants for the diene and the dienophile are about 100 micromolar. So very typical parameters for natural enzymes that are involved in metabolic transformations. Now, as I told you, there are no natural enzymes that do bimolecular Diels-Alder reactions. But enzyme designers have been very active to create such molecules in the laboratory. In black, you see a handful of examples generated by catalytic antibody technology. In blue and cyan right here is a ribozyme that also accelerates these kinds of transformations. In red is the computationally designed enzyme that I showed you at the beginning. At the beginning, right out of the computer, it's not very active. But again, we were able to optimize it by evolution. This black dotted line corresponds to the apparent transition state affinity for a natural Diels-Alderase that promotes a unimolecular reaction. We can't compare the rate acceleration or the effective molarity of these two systems directly, but we can look at the apparent affinity for the transition state. And this natural system exceeds all of these artificial catalysts. So the metallo-Diels-Alderase starts out sort of in the same region as these other guys. But through this process of evolution in the laboratory, we're able to create something that really is quite good. This exceeds the transition state of affinity for all of these other catalysts, including the natural enzyme, by 2 orders of magnitude. So I think that that's an unprecedented demonstration of the power of these technologies to generate new protein catalysts for useful biochemical transformations. This enzyme also, like the esterase, turns out to be highly selective. In this case, we have a diastereous selectivity. 1 of 4 possible diastereomers is produced preferentially, as you see here in this HPLC chromatogram. The same kind of conformational change that we saw in the esterase also occurred here, very early on in the evolution. We believe that that was dictated by the replacement of the original histidine that coordinated the zinc with a valine, and the conversion of the other histidine to a cysteine, which is a better ligand for the zinc ion. So it locks the system in place and preferentially promotes this transformation. In addition, although we don't have a ligand bound at the active site, we know that the binding pocket has been significantly expanded compared to the esterase. Here's the ester again, or the phosphonate bound to the esterase. And you can see that this pocket is much, much larger to accommodate the two substrate molecules and bind them so that they can collide in a productive fashion. So we're very excited by the system because we believe that it is going to be very useful for the purposes of divergent evolution. So you can imagine catalyzing a wide range of other transformations. In fact, we already know that our starting scaffold will do retroaldol reactions. It will do Michael reactions. It will do reduction transformations using Hunch esters, for example. So a huge number of different activities. They're all starting relatively low level, but we believe that we can improve them in the laboratory in exactly the same way that I showed you here. The fact that this scaffold is able to bind other metal ions should also expand the kinds of chemistries that we do. The original MID1 design binds copper as well as zinc. Our advanced, evolved variants will also bind other transition metals like rhodium and iridium. So we believe that there's a huge number of interesting transformations that we'll be able to access. To speed up evolution, we're going to need better and faster techniques. And I think here one of the most exciting opportunities is offered by droplet-based microfluidic techniques, which we've used quite successfully to optimize artificial aldolysis in the past. We've shown that this will also work with the hydrolytic reactions and are setting up now to try and take our esterase and turn it into a catalyst that really is diffusion limited. So in sum, we can make globular zinc bundles that are versatile catalysts. And these are really excellent catalysts for diverse chemistries. What's interesting here is that you don't need a lot of chemical information at the start of the design. You can rely on the intrinsic reactivity of the zinc ion to get you that initial starting activity that you need for purposes of evolution. And that gives us great hope that we'll be able to extend this approach to many other scaffolds. You may be aware of the enormous leaps forward in the area of protein design. David Baker and his colleagues over the last few years have been able to create atomically accurate scaffolds in many, many different forms. Before 2011, there were a handful of computationally designed proteins. Now there's just a zoo of structures, including Tim barrels, helical bundles, very exotic structures. Can we equip these with metal ions and exploit the resulting complexes to do novel chemistries that really nature hasn't seen before? I think it's a really exciting opportunity for the next coming years. So that's where I'll stop. It remains only to thank the people that did this work. Both of the projects that I told you about at the end were done primarily by a single graduate student, Sabine Studer, who's really a remarkable scientist. She had help from two master students, Anna and Sophie, who have now joined my group as PhD students, and some help from the chemist who made the transition state analog, and the crystallographers who determined the structures that I showed. This was also, as I told you at the beginning, a collaboration with Brian Kuhlman and Ken Hauk. And I'm really very grateful to them for their enthusiasm and hard work, and you for your attention. Thanks very much.
Thank you very much for an excellent talk, Donald. So we have room for questions. Many. I think you were first here.
So thanks for a very interesting talk. I have actually two questions. One is, can you understand now, once you have your starting enzyme with a low catalytic efficiency, and then you have some on the trajectory to getting the higher efficiency, what is actually happening? So why do these mutations contribute to
For example, in the case of the esterase, I think there are three issues. First, you have to create a pocket that will bind the substrate efficiently. And you can see that in the decrease in Km values over the evolutionary trajectory. Secondly, we tune the metal-ligand through these peripheral interactions, this extra hydrogen bond that adjusts the pK of the water molecule that binds to the zinc. And then lastly, we generate these additional catalytic interactions, for example, with the arginine that wasn't present in the original design, that helps to stabilize the anionic transition state. So those together, I think, make the difference.
But you have also, I think, mutations outside the...
Yeah, there are many mutations
so does
outside.
it change the flexibility, dynamics of your systems?
All of the above. I think those are all very important issues. Teasing their individual contributions apart, though, is quite difficult without doing... We have 20 mutations. We've mutated 20% of the protein. So it's a huge amount. But that kind of understanding will be absolutely essential as we try and go forward, because what we would love to be able to do is predict the final structure, not have to evolve it.
And the other one is, I guess you also, for these chemical reactions you want to catalyze, you probably also need some solvents.
I think all chemical reactions are...
Yeah, okay, sorry. But for this, I mean, the purpose of this is to synthesize some, or catalyze some reactions to get some new compounds. So I imagine that also solubility of your reactions can be an issue, so you might want to add solvents.
Sure.
So can you include this in the design process with your directed evolution?
That's harder, but it's well known that enzymes can also work in organic solvents, usually water-immiscible solvents. So you run the reactions in two-phase mixtures. And that should be also true here.
I think it was Vincent here. And then Rami.
Thanks for a great talk. I was just wondering whether you would be willing to make a general statement on how the work on catalytic antibodies, how you would rate that compared to the approaches that you were discussing. And
catalytic antibodies was a strategy that was developed because at the time we could not make protein libraries. Now we can make protein libraries. The problems with catalytic antibodies are manifold. You could catalyze many reactions with great difficulty, but you plateaued at an activity that was orders of magnitude below the activities of natural enzymes. The transition state analogs that you use are imperfect. You can't incorporate the catalytic residues that you need. You're selecting for binding rather than transition state, rather than reactivity. The binding energies available from the immune system are modest. And so you level out relatively low. What we found with the computationally designed enzymes and also these metalloenzymes is that we can go beyond by several orders of magnitude where we stopped with antibodies. So I think we've learned a lot from the antibody technology, and that's been fed into these kinds of designs. Many of the reactions that I've shown you were catalyzed by antibodies originally, but we've been able to go orders of magnitude more efficient.
Thank you for a great presentation. It's kind of covered what I wanted to ask already, but I have a more specific one. So, I mean, the design aspect works great. That's fine. It's quick. But then we're hitting a bottom when we go to directed evolution because it is not high throughput. So, I mean, apparently you show you cannot get away with it. But what is it that is happening? What will be the next step there to kind of speed up the process of developing them or, yeah, evolving them to the level that we want?
Well, there are ultra-high throughput methods. FACS is one. Droplet-based microfluidics is another, and genetic selection. And with these methods, you can typically look at 10 to the 8th variants in a single experiment. And we've shown that that's been extremely useful in optimizing autolysis, where we had climbed the mountain and then leveled off. And then using the microfluidics, which allowed us to explore many more synergistic interactions. Because typically in these experiments, we only let 1 or 2 mutations forward from one round to the next, because otherwise you dilute the advantages that you accrue. But using the microfluidics, you can look at simultaneously 4 or 5 residues mutated at a single site. And that gives you a chance of finding synergistic interactions that are beneficial for catalysis. And that allowed us to blow through the limit we had reached.
There is one over there.
If you repeat the mutagenesis evolution from starting a molecule, do you get a similar kind of final product?
I think that's impossible.
Okay. Yeah, but I mean...
Because the mutations are random, and
yes,
the sequence space is vast, even for a 100 amino acid protein.
I mean, I was more like, so the first round of evolution will determine the following sort of evolution. So at the end, it will end up in the same mechanism of increasing.
Yeah, but it's a branching tree, so
Yes.
it's an enormously branching tree. However, we have done the experiment that you have suggested with the aldolases. So we have an aldolase that uses a lysine as a catalytic residue. And we evolved this, and in the course of evolution, we removed the original lysine and generated a new one in the active site. And we were able to optimize that to very high levels. We went back to the starting design using the microfluidics and repeated the evolutionary trajectory. And we found different solutions. One kept the original lysine and optimized it, and another generated the new lysine or found the new lysine and optimized it. So in that sense, you're finding the same solution, but not not every single mutation is identical.
There is one more. I think we have time for that.
So, Cameron Allen, so I have a suspicion I asked you this question nearly 20 years ago, but it follows on from the previous one. Are we closer now to understanding in these different situations how much of the sequence function space we need to be able to access to get these optimizations and how much redundancy there is in the solutions? Or are we still just exploring little tiny parts of the landscape?
I think the latter. I mean, certainly these systems are incredibly redundant. If they weren't redundant, we wouldn't be here. I mean, the system has to be able to tolerate change. And if it didn't, a mutation would be devastating. But how much is actually contributing to the overall efficiency is a completely open question. And it's something that we're really keen to investigate using the computational and high throughput methods, because we can then explore different scaffolds. What's the best scaffold for this reaction? Did nature choose tin barrels for a particular reason, a chemical imperative, as it were? Or could any scaffold be as good if given the chance? And so we can explore those issues now, I think, in a more systematic way than was possible in the past.
One short one to that. So you can develop new catalytic functions by the artificial enzymes. Have you ever tried to, when you have your conclusion that works, have you tried to blast it towards the databases to see if something might exist in nature with the same activity?
So we've done this with the helical bundles, and there are many 4-helix bundles in nature, and you can find some overlap. In fact, the 46 amino acid peptide came from a natural protein. But the crossover angle is different, and the specific sequences are different. On the other hand, we hope to be able to generate lots of families, phylogenetic families, that would be useful to analyze in these ways. So thanks very much.
Thank you very much again. One big hand and a gift from the organizers.